Hi, my name's Melissa. I'm a fourth-year graduate student coming from the University of Virginia, and I'm really excited to talk to you today about how adipose tissue has heterocellular communication and how this can regulate lipid handling. Oh, I'm sorry. I have nothing to disclose. So here is a scanning electron microscopy image, and what I hope you can appreciate is how abundant capillaries are in adipose tissue. So the big purple balls are going to be the adipocytes, and the green little spidery looking projections are going to be the capillaries. And there's an abundance of capillaries in adipose tissue. Here is a transmission electron microscopy image showing a cross-section of those purple balls, and highlighted, highlighted here is going to be the lipid core of an adipocyte, and then this cell right here with the light gray membrane is going to be a capillary adipose endothelial cell. And what I can hope you appreciate about this image is how close in proximity these capillary adipose endothelial cells come to adipocytes themselves. So to really question and understand what these capillary adipose endothelial cells are doing and what their role is, I performed single-cell RNA sequencing. And I accomplished this by using a CDH5 Cre YFP mouse. So all endothelial cells are going to be fluorescing yellow. I took this mouse and either fed it a normal chow or a high-fat diet to induce obesity. And then I was able to flow sort out these yellow endothelial cells and perform RNA sequencing on the individual cells. So what we have here is a UMAP of all these cells that we sequenced. We were able to split them into different lymphatic clusters, or different clusters. First being the lymphatics, highlighted here in green. We had an arterial cluster, a venous cluster, as well as a capillary cluster. And I'm gonna focus here in on this capillary cluster here in pink. What I found really unique about this capillary cluster is that they are enriched for fatty acid handling genes. So fatty acid binding protein 4 and 5. Interestingly enough, FABP4 was originally thought to be an adipocyte-specific gene. It was actually the first Cre used. But here we can see it's not being expressed in adipocyte, it's actually being expressed in these capillaries from fat tissue. CD36 and glycoprotein 1 and CAV1 are also enriched in this cluster. These help facilitate lipid uptake into endothelial cells. And these genes are specifically upregulated in high-fat diet as opposed to normal chow. So what I've shown you so far is evidence of a unique population of these capillary adipose endothelial cells. And what I want to tell you about is how I found evidence of cellular contact between these two cell types. And so my hypothesis here is that this heterocellular contact between these two cell types can help regulate and facilitate lipid handling. So I performed an extensive EM study looking at the two membranes touching each other. So the first and most prevalent form of contact I saw in adipose tissue is between an endothelial cell and an adipocyte. And so EM images are kind of tough to look at as they're all shades of gray, but this darker gray is going to be the adipocyte membrane, and this lighter gray is going to be an endothelial cell. I saw some instances of pericyte adipocyte contact, so these are some supporting vascular cells. And I actually saw 0 instances of two adipocyte membranes touching each other. So this really places endothelial cells as a regulator of adipose tissue, as the two adipocyte membranes do not come in contact. So to really understand and recapitulate this model, I developed an in vitro co-culture system to really study this mechanistically. So I took a transwell, which is basically like a little cup insert with some holes in the bottom. I seeded these human-derived adipose stem cells, differentiated them into mature adipocytes, flipped it on over, and seeded human adipose microvascular endothelial cells on the inside. And then I'm able to treat with lipids or section this membrane to really ask some key questions. So here's a transverse view. So I just sliced that transwell in half. On top are these endothelial cells, on the bottom is the adipocyte, and these little holes here that you see are the pores. And here's phalloidin staining, so filamentous actin, just staining cell content actually comes through these pores, and I'll get to that in a minute. And if we look at the top layer, this is endothelial cells expressing the cadherin, that same Cree I used to sort these cells. And then on the bottom, we have adipocytes with a robust lipid expression. So when I say that there are contact sites or cell projections in these through these pores, when we think about cell contact in my lab, we think about connexins. Connexins are gap junctions that can facilitate these two cell types physically touching and communicating. So I looked for connexin expression in this cellular content. So these are the little cell contents in the pores. I looked for connexin 43, a very ubiquitous gap junction, and these little puncta here are little gap junctions, these little pores connecting two cell types. And this was really interesting because this has not been shown before. So what I did is I knocked down connexin 43 in the endothelial cells, and what you can see is I don't get any FABP4 expression. FABP4 is that fatty acid handling gene. So when I take away connexin 43 from the endothelium, we lose the ability to express these fatty acid genes. And the same if I do this in the adipocytes. So if I delete endothelial cell connexin 43, the adipocytes can't express this fatty acid handling gene either. And so what I really wanted to do is turn this in vivo. So I've generated an endothelial cell specific deletion of connexin 43. Here's a genomic excision gel showing loss of the connexin 43, a reduction in mRNA, as well as a reduction in protein. And so I'm really excited about this mouse. It's giving us an ability to look at this connexin and the relationship between endothelial cells and adipocytes in vivo in a living being. So first, interestingly enough, these mice do not have a difference in weight gain, which was surprising because I hypothesized that this connection is actually regulating fatty acid handling. So we don't see this when a mouse is fed normal chow or a high-fat diet. We actually do see an increase in fat pad mass, so the amount of fat that these mice have is increased. And there's actually an increase in cholesterol, most specifically from the LDL, that bad cholesterol, but otherwise these mice are metabolically healthy. And this is a theme that we've heard a little bit about today, which is resemblant of some lipedema characteristics. So in conclusion, for the first time, I've described heterocellular contact in adipose tissue between endothelial cells and adipocytes. Endothelial cells and adipocytes can contact each other, and this could possibly be regulating fatty acid gene handling. This heterocellular contact might be facilitated by connexin 43, a gap junction, and there is a metabolic phenotype in these endothelial cell connexin 43 mice. And actually ongoing, and right now, I've generated the reverse, so the adipocyte knockout of this protein. So if these two cells are coming together and each cell contributes part, I'm expecting that when you delete it from the adipocytes on this half, it's going to resemble what you delete on the other half. And with that, I'd like to thank my lab and specifically the Lipedema Foundation for funding a lot of this work. Thank you.